Hello everyone. Now today we'll talk about molecular basis of inheritance. What is the molecular basis of inheritance? Let's have an quick introduction. Inheritance means passing of characters from parents to children. These characters are carried by genes, which are made up of DNA. DNA stores information and passes it from one generation to the next. Who is the father of genetics? Mendel is the father of genetics, and he studied pea plants and explained how characters are passed from parents to offsprings. So we will learn about here law of dominance. When a plant or animal receives two different forms of a character, only one form appears, so the other form remains hidden. For example, the tall plant appears even when one parent is dwarf. Law of segregation. During formation of sex cells, the two forms of a character separate, so each sex cell carries only one form. After fertilization, they come together again. Law of independent assortment. Different characters are passed to the next generation independently of each other. One character does not affect the inheritance of the other. Let's see the deviations from the Mendel. Some characters do not follow Mendel's law. Incomplete dominance. Both parents' characters mix. Red plus white gives pink. Codominance. Both characters appear together like AB blood group. Multiple forms. A character has more than two forms like blood groups. Many genes involved. Some characters are controlled by many genes like skin color. Alright. Now let's talk about the chromosomes. Chromosomes are thread-like structures that found in the nucleus. They carry genes. Chromosome theory of inheritance given by Sutton and Boveri. Genes are present on the chromosomes. So what are the discoveries of the DNA done by different scientists? Transforming principle was given by Griffith, Avery, MacLeod, McCarthy. All these scientists gave the theory DNA is transforming material, and DNA is a genetic material given by Hershey and Chase. Double helix structure was given by Watson and Crick because the DNA looks like a twisted ladder. DNA consists of sugar plus phosphate plus nitrogen base. So the bases are A, T, G, C, or pairing is AT or GC. Types of DNAs. Prokaryotic DNA is circular and free in the cell. Eukaryotic DNA is linear and inside the nucleus. So A DNA is short and thick, B DNA normal form in the cells, and Z DNA is zigzag shape. Okay. So the pairing we already saw here. DNA consists of the nitrogen bases. So the four nitrogen bases full forms are A, adenine, T is thymine, G, guanine, C, cytosine. So adenine, A, pairs with thymine, T, and guanine pairs with cytosine, C. Okay. Now the ribonucleic acid RNA, it's single-stranded. Sugar is equals to ribose. Bases are A, U, G, or C. Types of RNAs. mRNA carries info from DNA to ribosome. tRNA brings amino acids to ribosome. rRNA part of ribosome. So DNA replication is a copying DNA. Semi-conservative. Each new DNA has one old plus one new strand. What are the steps? First is initiation. DNA unwinds at origin, helicase opens helix. Elongation, DNA polymerase adds complementary nucleotides. Third is termination. Replication ends, two identical DNA molecules form. Mutation, change in DNA. First is point mutation, single base changes. Substitution, frame shift mutation, insertion or deletion of base, shifts reading frame. Alright. So mutations can affect proteins and traits. Mutations can be spontaneous or can be induced. Induced by radiations or chemicals. So some mutations are beneficial, some are harmful. So genetic code is made up of triplet codons. The start codon, AUG, stop codons, UAA, UAG, UGA. So the code is universal and degenerate. Alright. So the start codon, let's understand the full forms here. AUG is adenine, uracil, guanine, then starts protein synthesis. So the stop codons are UAA, uracil, adenine, adenine. UAG, uracil, adenine, guanine. UGA is uracil, guanine, adenine. Stop protein synthesis. Sense codons code for amino acids. Nonsense codons do not code for amino acids. Okay. So transcription converts DNA into mRNA. RNA polymerase catalyzes the process. Promoter and terminator regions guide the transcription. Alright. Okay, the translation converts mRNA into the protein. The ribosome reads mRNA codons and tRNA brings the amino acids to form polypeptide chains. Alright. So the gene expression is regulated in the cells. Prokaryotes, operon model, for example, lack operon. Eukaryotic gene regulation is the more complex. So Human Genome Project sequenced human DNA that helps understand genes and genetic diseases, and the application are in medicine and forensics. So genetic disorders can be chromosomal or single gene. For example, Down syndrome, sickle cell anemia caused by mutations or abnormal chromosomes. So recombinant DNA technology combines genes from different sources. Restriction, enzymes cut DNA at specific sites. So plasmid vectors carry foreign genes into the cells. Some applications of recombinant DNA technology are in medicine, insulin, vaccines, in agriculture, genetically modified crops. So the gene therapy corrects defective genes in ex vivo outside the body, in laboratory, in vivo directly inside plant cells. So here is DNA fingerprinting. DNA fingerprinting identifies individuals, used in forensics and paternity testing. Relies on a short tandem repeat regions STR. So here is a summary. DNA is the hereditary material. Replication, transcription, translation occur. And genetic engineering enables practical applications. So the conclusion here is genetics explains inheritance after the cell division, and molecular biology is essential in modern science. So DNA technology benefits medicine, agriculture, and forensics. So this presentation explained DNA and inheritance, covers replication, transcription, translation, and mutations. Also that included gene regulation and modern applications of DNA technology. So here is a quick practice set. You can all go through all the question and answers here. See you in the next session.